minutes. Thank you so much for joining us. All right, we'll go ahead and get started. So thank you everyone for joining US HUPO today, this morning, this evening, this night. Today we have brought together the creators of 6 large public data commons that are being used in mass spectrometry community, whether that's proteomics, metabolomics, lipidomics, whatever omics you're doing. We've probably deposited our data in one of these repositories or multiple of these repositories. And if we haven't deposited that data, perhaps we are data consumers and we're computational biologists looking to pull that data out of these repositories. So today we're hoping to have a discussion about what's working with these data repositories, what's not working with these data repositories, what are some of the uses and the users that these repository creators built their databases to try to serve. We could also have conversations about who benefits, who should benefit from depositing data. How can we do a better job as data generators and as data consumers to make use of all this legacy data that we have from years and years of mass spectrometry experiments. So we're going to start off. Each of these 6 creators is going to briefly introduce themselves, introduce their repository, and then we're going to save plenty of time for questions, for discussion. So if there's anything that is kind of on your mind regarding data repositories, any aspect of data repositories for our 6 panelists today, feel free to use the question and answer box down at the bottom of your Zoom screen, Q&A, and we will be discussing all of those. If we have so many questions coming in, we'll put the panelists to answering those in text, but hoping to keep this more of a discussion today over whatever work you're doing in the background of this panel discussion. So first up today, I'll ask Yasit to come online with video. There he is. So we're going to start off with kind of the master repository, the one repository to rule them all. Take it away.
Okay. Okay. Thank you so much for the organizers for inviting me to give a short talk about PRI database. My name is Yaset Perez Rivero. I'm here on behalf of an amazing team maintaining PRIDE, led by Juan Antonio Vizcaíno. And I will talk a little bit about what do we think is the future for databases like PRIDE, not only for archiving database, but also to be hubs of mass spec data, evidences for other knowledge-based databases. But first, before anything, I would like to give a short introduction for those that doesn't know about Proteomics Change. Proteomics Change is a consortium created in 2014 of a group of databases. Most of them, you will hear about them today. That is integrated by JPOS in Japan, iProx in China, PRIDE in UK, Massive and Peptide Atlas and Panorama Public in US. You heard, as I said, a lot about most of these databases today in the webinar. But mainly the main task of Proteomics Change is define and coordinate a set of guidelines and rules about how to submit and deposit public proteomic data. This, I need to say, the first publication of protein exchange came in 2014. And I think from my point of view, personal point of view, this has been an amazing success in the omics field. I mean, we have right now more than 33,000 data sets. And we don't think about any publication nowadays that is not coming with their data public. And this is thanks to a lot of people who are behind the idea of protein exchange. And these resources who has pushed, also journals has pushed quite a lot, MCP, JPR, Nature journals that have pushed quite a lot to make public proteomics data mandatory to be public with the each publication. Protein exchange also is heavily involved in the development of a standard file format within HUPO. And this is a lot of work that goes behind the scene in the development of standards for storing data like MCML, all of them, very popular that we know all of them, most of the search engine and software tools they export into that. And the creation of this standard make possible to really, in the long term, being able to interpret the data. For a long time, the problem was that every software produced their own output, and those output has versions that they were not actually maintained. And when you have something like a database, like a protein exchange database that actually store the data for years, you really need that standard that can be kept and that has well formalized structure, and people maintain with libraries. And this is what HUPO PSI is all about. There is a lot of control vocabularies that are also maintained by HUPO PSI. In terms of PRI is actually has two missions. The first one is archival major mass spec data type and enabling the community the reuse of this data. And there is a second mission for PRI, is trying to bring that proteomic evidences and that proteomics information into a more knowledge base and integrated with more knowledge-based protein resources like UniProt, Ensemble, Expression Atlas, and actually bring that knowledge to a more biologists, no mass spectrometrists or experts in proteomics. And that's the second topic I will talk briefly. This is how we see actually the data flowing in proteomics. We have Proteome Central with the major archival of proteomics data where the data is submitted to it. And there is two major type of resources reanalyzing data systematically. Let's say we in the last 3-4 years, we have seen the emerge of new expression databases like Proteomics DB, MassQV, Massive Quant, and probably Nuno will talk about it, and Expression Atlas. And also we know all of them, all the sequence databases like PRI-Peptidome, Massive KV, Peptide Atlas, GPNDB that systematically reanalyze data. All this data is actually integrated in more generic databases, like for example, Open Target, Expression Atlas, NextProt, and UniProt. We can talk about how this integration should happen in a better way, or how people can contribute to this integration. As I say, PRI stored almost 29,000 data set. It's actually the major archivals of protein exchange. But it's not only about the volume, it's also, there is a lot of integration between the archival. For example, Massive replicates some of the data sets that PRI stored, which is an amazing thing because that means that we have replicates of data sets in different parts of the world. And there is a lot of coordination around where the data set goes. For example, for people who is trying to download data from China and Japan, which is far from UK and in US, people submitted the data to those repositories. There's a lot of coordination. The data continue growing. We can talk about that also, but also the size of the data. And that's from a lot of challenge for archivals. I mean, we have this small chart here when we see that, for example, every year, the amount of data set with more than 100 filed MS runs is continue growing in the same way that the number of data set. Finally, I will have two only slides about major things we did last year. The first thing is the creation of SDRS. This is a major problem in proteomics, the lack of experimental design and sample metadata. In most of the people involved, actually in this talk, also Mina and Olga were involved on that. The creation of a standard file format that can capture experimental design and sample metadata is really important to being able to reanalyze data for quantitative experiments. We can talk if someone have questions about that, but I will encourage you, if you're submitting data to Pride in the future or any other protein change repository, you should put your SDRF, which is the name of the file format. And secondly, I want to mention that it's increasingly more important to provide and develop cloud infrastructures, pipelines that enable the users to reanalyze data in the large scale environments. And we have developed something called QuantumS that people can start using, and we are using actively within the team. This is the team led by Juan Antonio Vizcaíno, and thanks for all the submitters of Protein Change and Pride in particular. Thank you.
Yeah, thank you so much for kicking us off. You've kind of learned this is, like the one master repository that kind of everything is getting pushed into. So now we're going to talk to some of these specific repositories. So next up, presenting very briefly, will be Nuno. We'll get his video up. There we go. All right, all yours, Nuno.
All right, thanks, Lindsay. Thanks, everyone, for being here today, and thanks for organizing this workshop. It's great to have the ability to reach out to people that we're trying to serve with the work that we're doing in the repositories. And I really like how it was framed at the beginning that, it's not just what people can do for the repositories, but also in many ways, primarily, what can the repositories do to help the community once all the data goes in there. So I'm going to give just a brief overview of how we see this at Massive, where we really focus on developing methods and ways to store the data to make it more findable, more accessible, more interoperable and reusable, or, in other words, just more useful for the community. So I'm going to give a brief overview of a number of things we're doing. I'll go fast over it because the goal is to give an idea, and then we can delve deeper into things in the discussion afterwards. So the first thing with any repository, obviously, you have to be able to submit data and find data. So obviously, Massive provides the ability to do that. We have close to 13,000 public data sets with many more still private, over 0.5 petabyte of data, if including those data sets. But really what we focus on more so than just the data going in and just staying in is how do we make the data useful over time. So as Yaset mentioned, we can import data sets from other repositories, and we do that. It's great that we have data, for example, at the Proteomics Data Commons, data Pride, and we can import from there into Massive. So even if the data set's not originally deposited in Massive, a lot of the things I'm going to talk about can be applied to those data sets by importing them, which we have automated workflows to do that. And one of the main things that have provided for people for a long time is this ability to convert the spectra into open formats, which tends to be a bottleneck if you ever want to reanalyze a data set and you don't have an instrument of the same kind. You may not be able to open the raw files, so we can do that automatically for you. And most importantly, we've always had this ability to reanalyze the data online so that you don't have to download the data to be able to look at it, to analyze it differently. And in many cases, when the data is submitted without the search results in an open format, you can actually generate new search results for that data and then see if you agree with it or not. And most importantly, I just want to mention here, you can upload your own. And this is something that we really encourage at Massive, is that if you reanalyze a data set, and many computational people here do, please make a point to share the results that you generated for that data set. And when you do, you can add the reanalysis and you'll get an identifier, RMSV or RPXD, and your results become attached to the data set and also publicly accessible. So another thing that we've done with the reanalysis of the data sets is we've built the massive KB spectral library, which, as far as I know, is currently the largest spectral library available for human data. And as it currently is, it's almost 5 million precursors for in vivo detection of proteins and peptides. And if including synthetic peptides, it's over 6 million precursors. And this covers over 55% of the human proteome, and it has pretty much every single protein is represented in there. There's a couple hundred that aren't, but the vast majority of them are. And this was derived from over 1 billion spectra and 30 terabytes of public data. So this is only possible because of the data that the community has shared. And one example of an important project that we have accomplished with this is this high stringency blueprint of the human proteome, which was marking the 10-year mark of the Human Proteome Project at HUPO, that really established, based on all the data that we have been able to reanalyze and share in the public domain, how much do we know about the human proteome? So this was a very important landmark paper that came out just a couple of years ago, and that's based on the data that we have available. So very quickly, you can search all of this data by sequence, by protein. You can find all of these spectra. There's about 1 billion PSMs that are now available if you want to look for spectra for any peptide or from any data set. If you want to have a protein view of the data that's available, if you're primarily interested in a protein, you want to see what peptides people have seen, how much coverage, you can use the Protein Explorer tool that we have that gives you a protein-centric entry into the data as opposed to data set-centric. And if you're really interested in looking at a particular spectrum, that might be an ID that you're interested in or the key identification in a paper, you can use the Universal Spectrum Identifier to look for that spectrum across all of Proteome Exchange data sets, if you go to Proteome Central, and also at Massive. And you can also use that to search for other identifications for the same spectrum. Has it been seen as something different? Is there a library spectrum for it or the protein information for that spectrum? This is all available at the click of a button. Also importantly, Massive also supports the GNPS repository for metabolomics data. And a couple of years back, we created this option to have a BLAST-like modification tolerance search against the whole repository. And at the time, this was published in Nature Biotech, and it took a few minutes, anywhere from 5 to 15 minutes to search the whole repository for one spectrum. So we found that to be exceedingly slow. So we've now developed a new way of doing this, where you can come to the repository with a spectrum, or you can use a spectrum from a public data set. And what this FAST tool will do is essentially search the whole repository or search the full spectral libraries for potential exact matches to your spectrum or modification tolerant matches for that spectrum. So this can be done in under a second for all kinds of exact matches, even when you're searching multiple billions of spectra in the Massive repository. And if you're doing tolerance searches, modification tolerance searches, it's under a second for libraries, and it's under a minute when you're searching the billion scale, repository scale spectra. And you can find reports of how many data sets have that match at what delta mass, or you can find how many of those spectra in those data sets are identified versus not. So it really helps you explore the data, in this case, in a spectrum-centric manner. So the final point I want to make is a very important aspect of Massive, which is the Massive Quant repository, where the goal is to share and support the quantification data and the statistical analysis of that quant data for the data sets, as opposed to just sharing the raw data and IDs, to also define formats and infrastructure for sharing the quant data, as well as to reanalyze it online. And one particular thing I want to mention here is there's this option in Massive Quant that you can go browse by protein, and then you can see how a protein has been detected as differentially expressed in all the data sets and all the reanalysis that are available. So you probably have seen volcano plots in specific experiments where this is a bunch of different proteins and each dot is a different protein. Here it's the same protein across different data sets. So you can see how significant it is when you see a twofold variation in expression. Is that common or is that rare? So overall, there's a number of different features that Massive and MassiveCon provide to keep the data alive, to add value to the data over time. But this really gains most when the community is more involved. So we'd love to hear from you how to make this more useful to you, and whether you have ideas to making it better, we'd love to hear about it. And of course, this is a team effort. Jeremy really runs most of the day-to-day operations of the repository, and we're glad to work with everyone else to put this in place and make it what it is today. So thanks for your attention. We're glad to address any questions you might have afterwards.
Thank you so much, Nuno, and kind of a great segue, because on your last slide there, you mentioned Mina Choi, who is our next speaker up, kind of building off of the Massive repository. Mina is going to be sharing more specifically about Massive Quant. There she is. All yours, Mina.
Hi. Thank you for the opportunity. I have a chance to present about the Massive Quant. First of all, I'm Mina Choi. I'm a principal scientist of bioinformatics at MPL department and in Genentech. Today, I will talk about the Massive Quant, what Nuno introduced. Luckily, I had a chance to work with the Nuno and then Yaset very closely a few years ago. And then I want to share what we have done so far. My presentation is more focused on the user side, Yaset and then the Nuno's group and then other the big groups working for the whole data repository, but I'm more focused on the user side, how I can get the benefit from this large data set. So the Massive Quant is what is Nuno show. This is the global resource for systematic sharing for all the quantitative mass spectrometry data. The overall, how we can start the project is that we have a bunch of the raw data set in the repository, but as a statistician and then as the bioinformatician side, we have a whole downstream analysis, and there's so many different combinations we can perform, and then all the results will be quite different. Then where we can save this one, how we can publish this whole the intermediate data set. Some people use the GitLab, some people use their own repository, but it will be really great if we can make a link from all the experiment design and raw file, and then publicly available. So that is a kind of our initial discussion, and then the Massive Quant is the developed. So Massive Quant is a really benefit for my perspective, because it has all the collection of the quantitative information, which is metadata especially, as Yas said, and I think it's pretty much every researcher now know this metadata, which is the description of the sample, and the experiment design is the most important to reuse the data set. And the Massive Quant also support the branch structure for reanalysis, as what the Nuno just show. And then another thing is they have a complete compilation of the whole public data set, including all the control mixture data, or the benchmark data set. This is also very benefit for the method developer, like computational method developers. And also the many tools nowadays start to inject the workflow, automatically we can run all the downstream analysis. This is also it can be possible because we pretty much all the data repository start to save all the metadata. So now currently, I just checked this number of the public data set in the Massive Quant today, so 72. 72, like overall in MassiveCon itself, more than 30,000 public data set there. But the 72 public data set have raw data and then all the intermediate data, also metadata and then quantification data and even statistical analysis one. So the full package are available for this public data set. And also it includes the multiple control mixture data, which is also quite important for computational method developer, which is fully documented, and then all the intermediate data set is available. So here is the some example you can see. When I start to look at, okay, I need this data set. Okay, let's go to the MassiveCon and then how I can find it. There are really good searching part. So here you can search by title, you can search by any number of the condition, any number of the biological replicate. For example, I want to compare ABC, the method, and then I want XYZ data set. For example, I want a big data set, for example, more than 100 biological replicate, or 2 condition, or multiple condition. You know, there are some condition I want to test it, and then here you can search everything in the data set. And based on this, I behave as a contributor. So now I'm working in the Genentech involved in many the biological study. And you know, Genentech especially highly recommend to publish the data set. So whenever we generate our data set and publish that, want to publish a data set, I use the Massive. So I am a data contributor for here. And also here, another things, you can also search it, upload it by. So you can just name your name and then you can get all the list of the data set you already uploaded. Or you can also find somebody else you want to find some other data set. So to submit the data set to Massive and MassiveCon, here is the list of the item, which is highly recommended. So here, what I also learned after I joined the Genentech, they generate so many different, experiment data. Then they have their own system. But this is the structure in the Massive and then Pride is very helpful to set up the infrastructure, data infrastructure in the company side as well. And then more practically, we have some whole tutorial on the documentation, how we can just submit our own data to the public data, public repository. I'm also as a consumer. So I am a statistician and a bioinformatician. I'm really interested to develop some method, computational method. And then I use the public data to show my method development. So for example, here is the list of the data set, which is already available in the Massive. And then here you can see how I use the branch structure provide from the Massive, and then here's the holder list. Everything is just completely transparent from the raw file to the whatever your p-value table. Everything is transparent and then reproducible. And then after I just use some public data set, I generate a new data set, and then I upload whatever our quantification data. Then I can become a contributor again. So here is how I use the branch structure, what the MassiveCon to provide. So here originally, raw files are there and GMX file there, but we can organize a container or reanalysis the part. I can use many different type combination of the data processing tools or the statistical method, and then everything you can save it in the one data set repository. So this is just a short example of how I use this data repository, but I'm happy to answer any question about discussion about how you can use this one, also industry level as well.
Thanks so much, Mina. Next up, we're halfway through these presentations. Next, we have Karen Ketchum. Awesome.
I think, thank you very much. Can you see my screen okay?
Yep, looks good.
Great, thank you. So I'm Karen Ketchum. I'm from the company ICF, and today I'm going to talk with you a little bit about our work supporting the National Cancer Institute and their proteomic data commons. This is a very large team effort, and it's really originating from the, I think, initial vision of Henry Rodriguez at the Office of Cancer Clinical Proteomics Research. So just a little bit about my background. I've been working with the Clinical Proteomic Tumor Analysis Consortium and their Data Coordinating Center for about the past 10 years. And some of this work is now being presented in the Proteomic Data Commons. Now let's see if that advances. And there we go. Okay. So I think I want to say first that we really are leveraging and following along a lot of the principles and proof of concepts that have been established by Pride and Massive. But like MENA, this is a smaller subset of data, and it's really focused on cancer proteogenomics. So the vision of the Cancer Research Proteomic Data Commons is to be part of a larger Cancer Research Data Commons, which you can see on the right side of the screen. And the Proteomic Data Commons is one element or node or data repository within there. Within our program, all of the cancer cohorts actually are analyzed with both genomics and imaging and many other platforms. And these data are stored either with the genomics data commons or the imaging data commons. So when the Proteomic Data Commons was established, it was set to be a warehouse for the raw data for the mass spectrometry, and also for process data, so that consumers could get peptides and protein information for those tumor cells. Its purpose is to make sure that the cancer cohorts are looked at together, and that the publications for these studies can be, and the data can be researched across individual elements. And right now it hosts NCI-sponsored research programs, including the CPTAC program, the International Cancer Proteogenomic Consortium, ICPC, and soon it will also host proteomic data from the Apollo research program, which is from the VA and the DoD. This data repository, as I mentioned, is fairly new. It was established in 2020, and it took over from the original NCI CPTAC proteomic data portal that was established in 2012. So I'm going to walk you through a couple of the screenshots for users that they might look at when they're looking at the Proteomic Data Commons. And so when you log in or go to the website first, you'll find this first screen, and it's probably familiar to many of you because there's a theme, a commonality between the Proteomic Data Commons, the Genomic Data Commons, and the Image Data Commons. And so you'll see here on the left, there's a depiction of the human body, and then you have selectable elements for each of the cancer types that have been studied. In parallel, on the right-hand side are a list of the cases by disease type. So this is the initial draft dashboard for the PDC. And then if you look at the top, there's a navigational bar in the white menu where you can look to browse and look for analysis, data use, publications, etc. If you were to go to this navigational bar and use the browse button, you'll come to a screen that looks like this. And here you can filter the data based upon many different choices. You can do it by the primary site of the disease, the program, analytical fraction, etc. So here, this picture here, I've just selected the CPTAC program, and what you find in the center is a display of the different disease types that are from this program, experimental types, and analytical fractions. And then below, there's a list of the different studies. In the PDC, the term study is used for a collection of data that all has the same type of, it's on the same cancer cohort, and it has the same methodology. So we will call, there might be a publication that has proteomics, phosphoproteomics, metabolomics, et cetera. And each of those will be a separate study, but they will be linked together. So if you come to this study list below and select on one of the studies in the PDC under the PDC ID, you'll come to this type of screen that provides additional information about the specifics of this study. So here you have on the left-hand side the number of cases or individuals that were evaluated. The aliquots indicate that for many of the cases, we have multiple tissue samples. We may have tumor and normal samples. We may have tumor and blood. And then there's a summary information in the center, and then links to be able to get to the various data files. You can also scroll across this top menu and look at the protocols, experimental design, get the clinical data, and also information about the biospecimens. Not shown here are some of the links to the analysis tools and APIs. The Proteomic Data Commons also has a separate portal for data submission. And right now we're supporting NCI program submissions, but and in the proteogenomic area for cancer, but we certainly, depending upon NCI's spectrum, welcome people to look into submitting their data to the portal. And so submitters can find a login area, data models that are shared between the CRDC nodes, and then we work on metadata capture and harmonization. Presently, there's a wealth of data within the Proteomic Data Commons. We've had many flagship studies. There's 18 proteogenomic studies for cancer from CPTAC and ICPC that goes through colon, kidney, ovarian, lung, and many other cancer programs. There's around 29 terabytes of data, and we have an international community that's downloading the data. And it's exciting to see that places like Massive also have copies of these data, because I think the more people that share it and the more tools that look at it, the wealth of the information that will grow. And then finally, so I've talked a little bit about there's a public website, there's a data submission portal. I haven't talked very much about the fact that it's within a cloud environment, it's scalable, it's FSMA and FedRAMP, but this also goes toward the goals of NCI to have an interconnected cloud working space for individuals to explore cancer genomes. And then finally, I wanted to emphasize that within the Proteomic Data Commons, if you look under the More button, there's a wealth of information about how to use this data repository. There's a large section on frequently asked questions. There's information about how to use the API. So we always have two different communities. We have people that want to use primarily APIs and programmatic access to the data. And then we have other community that work through the user interface. And then I want to also note that there are release notes there for each of the data releases as well as software releases. So I think, or at least I hope, this gave you a little bit of taste of the information that's available in the Proteomic Data Commons. And it's also exciting for me to note that Dr. Bing Zhang, who's also talking here, was one of the major contributors to data within this repository. We look forward to your discussion, and thank you for your attention.
Thanks so much, Karen. I definitely learned something new already today, because I was still thinking about the CPTAC portal. So now I know I'm 2 years behind in my repository knowledge.
Yeah, that one's retired.
Whoops. Next up, Bing Zhang. Awesome. Looks like you're ready to go.
Yes, first I would like to thank US HUPO for including me in this panel discussion. My name is Bing Zhang. I'm a professor of molecular and human genetics at Baylor College of Medicine. So my lab is interested in computational proteomics and multi-omics. So our research is supported by, as Karen just mentioned, the NCI CPTAC, as well as CPRIT and the McLean Foundation. So we already heard a lot of, good things about the generation of the mass spec data, and also on the other side, not only mass spec data, but other omics type of omics data and multi-omics data are also available. And the data repositories are available, as we just heard, all these excellent resources. that allow user to reuse the data. But in reality, I think that most of this data still flow to the small number of computational biologists, but the broader research community, like the biologists and the clinicians, it remains difficult for them to access and use the data. So one research direction my lab is taken is to ask whether we can develop tools that will also enable the data to flow to the general audience to allow them to also use the data. So I'm not really like a data repository hoster, but I think we are more like a broker of these large data repositories. Our work is actually built based on the very hard work from the Pride, Massive, and PDC, but we're trying to build a gap in a way to connect this data hopefully better to the end users. So one tool, for example, we have developed is called Linkomics. This will allow users to not only access, but also analyze TCGA and CPTEC multi-omics data. The tool has a total of data on 32 TCGA cancer types and 10 CPTEC cancer types, data from more than 20,000 patients coming from non-omics platforms, as well as clinical data. It's not only a database with all these processed data matrices, but we have tools that can, online tools that can allow users to, just through click on the buttons to perform Bayesian and cross-omics association analysis and perform multi-omics and the pan-cancer kind of integrative analysis, and then use pathway and network analysis to interpret the output from these results. So this has been reasonably well used. Since the publication in 2018, there are already more than 67,000 users and more than 1,000 citations. So some users like this because it's flexible, provide different analysis, a lot of data and analysis capacities. But still, other users don't really like this, right? It's flexibility usually also means a learning curve because you want to learn all what it can achieve, right? Some users prefer a Google-like interface, so they can just type in something and then they get a lot of information on that particular thing. So to address the needs of that type of user, we also recently developed the Linkomics KB. So aims to use the proteogenomics data to develop some knowledge base for human cancer proteins. So this is built upon the CPTEC pan-cancer working group harmonized the data from all the 10 cancer types. But rather than let the users to perform analysis online, we did all the computations on the back end and then store all the results in MongoDB. And then we provide a very easy or to use web portal. And users can just type in a gene name or phenotype name or mutation, and then get all the pan-cancer multi-omics information for the whatever they are interested in. So these are just some of the visualizations provided in the web portal, which is to be released very soon, hopefully by the end of this year. So another tool I would like to briefly mention is PepQuery. This was inspired by the BLAST, but basically allow users to type in or provide a peptide sequence and then query a collection of MS spectral data and to basically identify the peptide spectrum match. I think Massive now also has capacity to do some of this, but this was published like a few years ago, but it was not really, it was difficult to scale up. But more recently, and Bo used to be in my lab, he come up with a new way to index the data to speed up the query and reduce the storage. And applying the new indexing method, and he basically indexed all the more than 1 billion spectra from 48 large data sets, downloaded, of course, from Pride or Massive. And then with this new capacity, users can query one sequence against all this data and to answer questions like to get support for novel peptide predictions or validate in peptide protein identifications from database searching, because this is using a very different algorithm. And then to prioritize tumor-specific antigens, hunt for missing proteins, and select a proteotypic peptide for targeted assay, etc. So I will stop here and thank you for your attention. I would be happy to answer any questions after all the presentations.
Thanks so much. This is our last presentation coming up. Keith Bradford going to be talking to us about Panorama, which I see we already have some questions about. All yours, Keith.
Thanks, Lindsay. My name is Keith Bradford. I'm from LabKey Software. We're responsible for the Panorama application, and then specifically we're talking about Panorama Public today. Give me just a sec. Okay. The main portion of what we have is just really answering this question for your next paper. How, and you've used Skyline, how can you make that publicly available? And really to understand that, you've just kind of a little bit of background of the relationship between Panorama, Panorama Web, and Panorama Public. Panorama is an installable software that is developed by LabKey. Panorama Web is then Panorama in the cloud that is hosted by the McCoss Lab at University of Washington. The researchers can request free projects to then use Panorama in the cloud. And then Panorama Public is then a way to make your data specifically that is matched with manuscripts available to the public for consumption. Panorama Public has a variety of different uses, and but really focused on Skyline and being able to upload and organize that data. And really that organization part, I think, is one of the strengths in that you are able to work with your data, you're able to upload your raw data and your analyzed data, and then organize it and have some control over the presentation of the data before submitting the whole folder to become publicly visible. Once submitted, that data is initially can be kept private. It's not initially available to the public during the review process. Then you can make edits and changes as necessary by the review, and then make that publicly accessible for the whole group. Additionally, during that submission process is Proteomics Exchange ID is associated with that folder. Okay, there we go. The process for submitting data is really just quite simple. You request a free project, you're able to upload your Skyline documents, and then your additional raw data, and then add whatever metadata that is appropriate. And this is really the part where you can then organize and choose how you would like to present the data. There's a wizard that kind of walks you through the different forms. You can just fill in the required portion, and I can show you an example of what that'll look with just kind of the generic. And then you can also go above and beyond and put in some more customization to add visualizations or additional information, or make additional files available. And then choose your link and submit the data to become publicly available. This just shows that once it's in Panorama Web, the submitter can work with the data and do whatever they want. Once that is then copied over to Panorama Public, it's a read-only format, and so the data is going to then be static and available. And then you can have your various visualizations and being able to go through everything. I did want to share just really quickly. I saw that there were some questions about raw data being available. So just to show an example of a project that is now available. This is kind of the generic where you've just kind of filled in the required fields. Raw data is then available in this upper right tab over here to then be downloaded and used. And then just an example of being able to organize the data and give some additional information for users is here you can see they've done kind of the generic portion at the top. And then additionally, they've started adding additional information and subfolders to try to make it a little bit easier for the users to access the data and understand what was going on with the project. And then last, I just want to show a little bit about the searching capabilities. This is just the general dashboard, and then users are able to search from a variety of different methods depending on what they are looking for. And then this is something that is under active development to make the user experience a little bit easier and more straightforward so that they are able to search and find data a little bit more easily. Thanks so much for the time, and happy to answer any additional questions.
Thanks so much, Keith. That is all of our introductions. So I think we're going to bring all 6 of our panelists back up, and we have got about 45 minutes to grill them with all of your questions. I saw we had some questions in the Q&A that we can bring up again as part of kind of a discussion. But if you have any others, feel free to drop them in, and we will kind of open up the floor to see what people are saying. So one of the questions I thought maybe could kind of be addressed to all of the panelists. Dana Robinson asking about replacing MZML file formats with something HDF5 based. So file formats in your repositories, is it best to keep just one file format and force everyone to upload the same file format so that tools based on your repository can do easier data pulling out of the repository? Or is it best to keep it kind of open and allow people to deposit any kind of data into the repository?
No, do you want to start or?
Either way, I can start with it. So one thing to keep in mind when it comes to this question is that one of the main goals in proteomics change is that we want to connect the discoveries that the data supports to the raw data. So from that perspective, it's always important to have the original raw files because they contain additional metadata. And when you convert them to open formats, they may, there may be centroiding, there may be other kinds of processing that transforms the data and loses some of that information. So it's always important to have the raw data. And going beyond that, if the data was analyzed after conversion to MZML or to some other format, if it was a part of your data processing pipeline, it's good to also have that because your conclusions may depend on what happened in that conversion step. But otherwise, we at Massive, we have the ability to convert the raw files into open formats to facilitate reanalysis because most of the open source tools will work with the open formats and not necessarily with the raw formats. So it is important to have them in open formats. And for us, the main goal is that it be usable for most tools. It's great if we can use a more compressed format. We know we have hundreds of terabytes of storage, so we know what it is like to have to deal with storage issues. But most importantly is that the data be usable. So that's what we focus on. I don't know how many tools would currently support these new formats, but certainly as they become more supported in the tools, it can make sense to switch into those formats.
I would like to add only that at the very beginning, like 15 years ago, we used to store like intermediate formats, not the raw files at the very beginning. And the decision was to say, okay, let's start from the provenance, from where it's coming from the instrument. And that was a clever decision because over the years, we have seen that the file form intermediate formats or standard file formats even evolve and you really need the original source. And that's what has enabled us to basically replicate the original results and so on and so forth. As Nuno say, as soon as you put the software in the middle, you really will do, like, you really do some transformation, and it depends on the software parameters and so on, how this spectra is transformed. Then raw files, I think, is my opinion, but also I think is the general consensus in proteomic change, will be forever stored in the database. Then what else after that? Actually, there is a really nice trend within the last couple of years or 3 years, I think, and it's a big change in proteomics, which is the APIs to interpret those raw files are available. And that's a change a lot the field. We have now open source APIs that actually can read raw files. That didn't happen before, and it was really difficult. And that's why we really need a lot of file formats in the middle, MGF, pick list files, MCML, and so on and so forth. And I hope that will be the trend. The trend is like we have more supported libraries from the instrument providers to read different versions of their own files. But coupled to that, I think standard file formats can evolve for intermediate. As Nuno said, we have huge amount of data that it doesn't make sense. For example, when you do a reanalysis, to actually keep track of the raw file, it makes more sense to keep track with a file format, MCMLB or something else, that is smaller, more efficient to transfer, and so on and so forth. Yeah.
Yeah, I love that. The idea that with these API tools for even just downloading data out of these repositories to pipe them in to reanalyses makes it just a lot easier for people who maybe aren't super computational pros to now take advantage of all this legacy data that we have acquired as a community, either in these kind of global repositories like Proteomics Exchange or Pride, and even in these more kind of niche repositories like the NCI Cancer Commons. Getting more questions kind of flood in. So we'll start with a broad question for all the panelists. What is the next big challenge for data repositories? For example, will we have integration between proteomics repositories and other repositories that focus on genomics, transcriptomics, other types of data? It seems like quite a big challenge to me. Have we fully embraced the challenge of even just proteomics data repositories? Are we ready to branch out and integrate other data? Maybe we'll start with Bing Zhang this time.
Yeah, so I think that's an interesting question. Of course, I think there's still a lot of things to do, just a lot of remaining challenges in proteomics. For example, even proteomics and getting all this PTM data, right? How to handle all this, and even how to analyze the isoforms. I think there are a lot of unsolved problems in proteomics. But in a larger context, I think one thing I always ask myself is that proteomics has gotten a lot of development during the past 10-20 years, right? I think we are in this field, we feel this like a rapid change, and we are very excited about all this. But if you go to for example, AACR or other if we go outside of the proteomics community, go to any cancer conference or any other, like the disease specific or biological development, or even just molecular biology conferences, I think people are less appreciative of the proteomics, in my view. So, for example, we worked in the CPTech, and everyone is very proud of what we have done. We did even publish a lot of papers in high-profile journals, but most cancer people don't. Sorry to say, not a lot of people are not aware of CPTech. Then of course, everyone aware of the TCGA. So I think one thing I'm thinking is how can we really go beyond the proteomics community, make all this data, try to make impact directly to the general biology audience. That's why, I mean, we are actually trying to develop some tools to make proteomics data useful in a way that anyone can use it. Not just, I know there are a lot of challenges for the analysis of proteomics data itself, but I think there is also a challenge how to make this also available and appreciated by users outside this community. So I think that hopefully I think it's improving a lot. Now, I think, I mean, a lot of people, don't get me wrong, I think a lot of people, proteomics is gaining more and more attention now, but I think we just need to do more.
So if I can go next, I think this is an excellent question, and I'd like to add a few comments. I think that one of the biggest challenges that the repositories had from the beginning was to prove to everyone that it's possible to have an infrastructure for data sharing, that it is possible to provide the service to the community. And I think that has been a huge success, like Yaset mentioned, that now it's just considered a given that this is going to happen when papers come out. Now, I agree that the main challenge that the repositories have is, now that we have the data, now that this is commonplace to share the data, is to answer the question, how do we make this useful for everyone? How do we help drive this process? And I agree with Bingjiang that we have to look outside towards people who are not in proteomics to use proteomics data, but I also want to look inside and make the data more useful for everyone who's generating data, for everyone who's running experiments in our field also. And I think we have still a steep hill to climb here. And one of the things that Bing and Karen are very fortunate to have in the context of the CPTAC program is they have fantastic metadata. And that makes it possible to do a lot of things that we still cannot do for most of the data that is shared publicly. So Yaset mentioned this earlier also, and I want to reemphasize this point. Please submit metadata with your data sets. If we want to do anything that's biologically meaningful, it cannot be done without the metadata. It's even more important than the search results or anything else. If we don't know how this matches to samples and to phenotypes, we really cannot link it to anything else meaningful other than just, the basic searching of sequences and whatnot. So I would say that's probably the biggest next hurdle that we need to overcome as a community, is make sure that we have proper metadata for the data we're sharing so that we can then do all the other things that everyone else wants to do.
So maybe I can chime in here a little bit. I think that this is kind of the vision of the Cancer Research Data Commons, and that each of these interlinked repositories within NCI will build upon each other. I think that there are some real barriers to entry. So we have a community that's very well skilled that can move easily between these repositories, but I think it's very important to continue to develop educational, informative, and access tools for people that may not have, as they have the questions, but they may not be able to easily navigate between a lot of these repositories, because that's a lot of the barrier that actually I see. And I think people have a real interest in using data. And also, so for example, right now we're at this challenge of having the proteomic data, but then also having to start to manage metabolomic data, which is actually similar, but actually not. So, one would anticipate there'll be in the CRDC at some point in the future, metabolomics data site, but that's kind of how we see it evolving. But I think that those two things also, if you're continuing to keep the raw data, I think you have the ability to move it forward, as techniques and methods increase.
Now we'll do Yaset and then Bing.
I think this is a really important question, and I think it's actually the future of most of our archivals are, how do we make this data from the archivals available for mass spectrometry for our community? I mean, mass spectrometries and researchers that knows proteomics generate this data, but we want to make this data available for the other community. And that's the challenge, and that's what we want to work with. Mainly, for a long time, all the work was about, a lot of work on standardization was about how we share the data ourselves. How, for example, we share a peptide. What is a peptide for us? How do we represent a peptide? What is a score of a peptide for our PTM, and so on and so forth? As Nuno said, if we want to reach other people, and that's why we actually pointed again back to that, there is a lot of effort that needs to be done to describe our data at the level of the metadata, the biological insight, where this sample come from, how the experimental design was created, how samples are combined, what is this patient, where is this tumor, size of the tumor, this kind of the metadata. We didn't care before as a community as much as we need to care if we want to bring this data to others. And that's actually the major challenge for us. We are in Pride, Nuno probably also is doing it, and Proteomics Exchange, re-annotating data sets ourselves, putting out for the users to know, okay, this is the way you annotate data, reanalyze it, and put in the final expression data, PTMs, and so on into biological context. Okay.
And Bing.
Yeah, I just want to re-emphasize the importance of the metadata, because I think what makes the data useful outside the proteomics community is probably because of the metadata, because people want to ask interesting questions. That is always related to metadata, I think. So, but I think the challenge we talk about, I think probably everyone would agree on the importance, but I don't think this is a new challenge. The question is, I think, like Nona said, Karen and I will work in CPTech because these questions are very well defined. I mean, at least it's still broad, but it's cancer, and there are some important questions and important sample attributes that we want to connect. It's already kind of clearly understood, right? But if we go beyond the cancer scale, like a massive or Pride, because you guys host all kinds of data from all model system, all like just anything, right? And then how can even for something like GEO, right? Probably a lot more people work on that for much longer time. I'm not sure there is a really good way to organize all the data in the gene expression omnibus. So I think the question is, how do you guys or anyone has any suggestions on what would be the best way to standardize, or is there really a hope to standardize the metadata?
I could just say very quickly, and I posted as a response to one of the questions, that there is a format at the STRF and MageTab Proteomics that was published recently exactly for this purpose. It was based on the MageTab format that is used for, I believe, Expression Atlas. Yes, correct me if I'm wrong. That was also similar to GEO, same kind of thing. So, but extended for the additional things that we need for proteomics experiments. So as for them, this is a mapping of the raw data to samples. It does not describe the statistical analysis on those samples, like which conditions did you compare against what conditions and whatnot. So there's still additional work to be done there, but I believe it's the closest we have to the level that you would also have in CPTAC, which is describing the samples and describing how that matches to the raw data. So this was published a year ago now, actually almost to the date today. So this was a very important step for being able to move forward in a community, to have a common language that we all speak. So we have that now. So now we need to start populating. There's a lot of work ahead to actually start using it, but the format exists, and I can post it again just to make sure everyone's able to see it.
And Keith.
Yeah, I know everybody has kind of talked about kind of the importance of metadata, and often I'm working with groups that are kind of working before submitting their data to a repository. And one of the things that the challenges that I hear most frequently is how can we carry that metadata throughout the steps of our process as files go through different file types, different tools passed to different groups, just how can we preserve that data throughout the steps of our process? And so that's something that we're working on, and I guess trying to build tools to make it so that it's efficient to capture that metadata. So then when it comes time to put it in repositories, it's not an onerous task to then go back and try to gather what you have.
Yeah, definitely. I'm a big advocate for collect that metadata and record it as you're doing your experiments rather than you're looking at your list of hundreds of raw files and trying to go back and remember what exactly raw file, 22 10 13 LKP QEHF 11111 was. Kind of on this topic of metadata, have a next question that I'm going to kick to Mina. For large-scale proteomics projects, samples were probably processed in different sites and different instruments for the data acquisition. So what might you recommend for mitigating possible batch effects for reanalysis?
So thank you for this question. It's the batch effect. Large scale normalization is a really key word, right? This one is really common. Even we don't need to go to the large scale. Like we can just, even small data sets, small two data set. If you have a two different time point, we start to have a seed of batch effect. Very practically, sometimes we have to give up because there's, this is not well designed. So I always strongly recommend to talk with the statistician before you design this large scale one. We have to consider a lot of the control, normalization, how we have to put them before. So now I think it's practically, technically also, it's really hard to merge large data set is already performed without the considering like this normalization, etc. So I cannot tell any general one solution for this one, but yes, but I strongly recommend, even you don't need to go to the large scale, even small data set, always, consider this one before you, perform the experiment. That is only my recommendations.
Yeah, any other thoughts on how to integrate data from multiple experiments in any of these repositories? Definitely sounds like metadata is the key for sure.
Yeah. We assume the metadata is there. So.
In Pride we have a
Go ahead, yes.
In Pride we have a mechanism already in place where if someone submitted an experiment to EGA, ENA, or one of the genomics or other omics archive, it can provide the accession of the data of the raw data in the other side. And that's a key point because you know where the data is, and then you can have multiple accessions in the same place. That's one thing. And going back to the file format, that file format was designed thinking on the other omics. I mean, we didn't design that file format SDRF from scratch. We actually looked into the genomics field and also transcriptomic field, how they were sharing their data. We have more complexity because we have more multiplexing that then we have fractionation also that most of the other omics they don't have. And we have different kind of experiment, turnover experiment. We have protein-protein interaction experiments and so on and so forth. All of that complexity for 1 year, we try to capture in the file format that is actually compatible with other omics field. Then I think we are in a really good position right now to start writing, giving training around that and making better the integration actually.
All right, so I could give a quick note on this. So one thing that we do at Massive, we do a lot of reanalysis, and we built, for example, the Massive KB spectral library from over 30 terabytes of data. So we've definitely integrated results from many different labs, many different instruments, many different kinds of data to do so. And in those cases, I see where Mina is coming from, because she's thinking about the important questions, which is how do we use the data to answer biological questions. But there's other things you can do earlier in the process that there are other simple guidelines that you should keep in mind while doing those earlier steps. You should make sure that your analysis always is FDR controlled in the context of the initial experiment, initial data set, initial design, initial samples. So you need metadata for that. And then when you integrate, you need to control the error rate of that integration also. So correcting for batch effects at that level may or may not be possible. Some things, if it's just instrument variability, random background noise, they're easier to correct for. But if it's poorly designed, as Mina said, you might just have to throw it away and not be able to use it at all. So there are simple things that can be done, but unfortunately, in some cases, it's just not enough, and you just have to not use it, unfortunately.
Yeah, I think there are two levels of batches in a way, right? One is a batch in a designed study. Even in design like CPTech study, there are already batches in a particular like head and neck study, right? I think the batch information should be required as metadata and to be submitted together with the data. So I'm not sure even, for all studies, do people submit the batch information? Because otherwise, reanalyzers will have no idea where the batches are, right? I think that is important. But then beyond the individual study, then you want to integrate multiple related studies. If the metadata can point you to the right way to, these can be integrated. I think then there are two options. One is, of course, if you can figure out a way to normalize, integrate them, then you can gain power by combining the data at the raw, not the raw, but the data level. Another option is just to perform analysis separately, appropriately for individual data set and integrate at the results level. That's one way we typically use to get around of potential, batch impact from different studies. That's, you are probably going to lose some power, but then you are not affected too much by the potential batch effect across studies.
Yeah, I want to jump into that part. Bing just said something really, really important. For a long time, everyone who is reanalyzing data and putting data out there has been thinking on the integration part of it. Meaning, I take, I don't know, 10, 20, 30, 40 experiments and try to merge all of them. And then, of course, when you have multiple meta-analysis, then you start raising issues around batch effect correction. You need to quality control of some of the data set and so on. But if you go to the field of transcriptomics or omics field, there is a lot of resources that they're focused on independent experiments to be out there in the resource. Then you have the integration of multiple data sets all together, like a view, let's say expression profile for something, but you also have independent data sets, when you can control the batch effect correction at the level of data set, and they are presented to the user. And this is actually, if we think more in this direction, we may end up by having multiple resources in the future, when you can, in some of them, they can actually deal with the batch correction effect and other effects of data integration, like FDR, control of large data sets and so on. And you might end up with other data sets when you have different views of different data sets. And I think this is a really good point for the future.
Yeah, this talk of batch effect correction really ties in with the metadata. And we kind of mentioned, at what level do you do batch correction? Do you do it at the raw data and do a reanalysis of all the raw data, or do you do the processed files? And I think that gets at Mike Riffle's question. To what degree does being able to access raw files in the future continue on the continued cooperation with vendors? So being able to access raw files, do the vendors have to still be involved in all of these discussions? Or if we lost raw file access, is it better that we use these repositories to submit data in open formats, which should always be vendor neutral?
I can jump on that. I think it's since the creation of containers, Docker containers, we have always a way to convert the raw file because we have an static version of a software that can actually translate that into an MCML or something else. I think it's a good point when the data become let's say too old, but still interesting. And that's a good point, because some people think, okay, old data is not interesting. There is a lot of really old data, really, really interesting. Tissues that hasn't been explored again, or tissues or data set that has a lot of deep. However, I think with the creation of containers and containers around software, you can see right now that some specific software, like for example, thermo raw file parser, which can translate thermo raw files, or for example, Proteo Wizard can be containerized, and then you will have old versions of the software that can interpret and translate to MCML and standard file format. I think it's a good point that when we said before, raw files, standard file format, we are in that business. We actually will continue developing standard file format. We think it's important to share data. But I think our point is that now we have a vision that involves more things, like APIs from the instruments, the native raw files, and the standard file format. It's a compendium of things, yeah.
So not a problem, because now we have Docker containers. So we should always be able to convert our files into something more useful besides a vendor proprietary. Is there any concern about the fidelity of storing data in a raw versus an open format? Should we be concerned at all about any metadata that might be in the raw file itself that doesn't get saved into a converted file?
Yep, that's why we would like to have the raw data always. And actually, in regards to, we were just talking about batch effects. Oftentimes, you can even detect some of that from the raw file because it tells you the date when it was acquired, for example, and that alone can tell you if you did all your cases before all your controls and essentially messed up the whole thing to begin with. And some of these things may just not be captured in the open formats. So it's always important to keep it. And I don't think we've ever lost access to any raw files. So hopefully it'll continue to be the case.
So kind of going back to this idea of comparing data, using these repositories that we compare data across experiments, with quantitative data comparison, what are the criteria that you would have in mind when assessing whether a data set could be compared or not? And I think, yeah, Mina, this is perfect for you.
Yeah, so to clarify the question, I'm thinking about this scenario. So for example, data public repository, we found 2 experiments. They look like they're quite similar. Want to compare any output from there. So this is actually comparable or not, right? In the final p-value table or something, we can compare the result or not. In that cases, first, just come back to the metadata. So I want to come back to the metadata. Why? Because I want to define the scope of the comparison first. So for example, okay, so these 2 experiments, they have the same condition, right? For example, one condition healthy and then disease 1, but the other one is disease 3 and then disease 5. They are completely different, scope. Then we have to define, okay, what kind of the things that we can do this, right? Or control and then disease and then control and then disease. And then they have hold the metadata for the patient as well, they have a similar subtype or something like this. So I want to define the scope of the conclusion, how we can make it. I feel like the more equipment or acquisition type is not that much important than the metadata. For example, something is from the DIA, something from the TMT is fine. We know that is a difference about the acquisition, but first look at the metadata, including design and then all the instrument, define this is the one can do, and then based on this we can say, okay, this is a difference, probably based on this one or probably based on this, but without to consider everything, just do and then different. Okay, why this is different? This is really hard to compare the result.
Yeah, seeing a recurring theme here with metadata. Big, how about you?
Yeah, actually, I want to touch upon something slightly different. Metadata part is very important for this. Like Mina already said, I mean, if they are not comparable, I mean, the question is not comparable, you should not integrate the meta, right? But another thing I want to touch upon, especially, I mean, we are now talking about the quantitative data, right? I think another thing you want to think about is the quality of the data. Do you really want to integrate that data? I mean, I think the quality is very important. I think in Proteomics has been heavily focusing on the quality control for the identification. I think FDR is not ideal. I mean, there are problems, but I mean, I think it's okay. I mean, at least we have one very well established way to, I mean, benchmark or compare the performance or things like that. But I think for the quantitative data, it's actually very difficult to assess, I mean, whether the quantification is good or not. But if you think about it, after you identify a lot of different steps after that, from getting the quantification and then normalization, batch effect removal, you would think all these are good, but it may, I mean, if you use very different normalization or different batch effect removal, you may end up with completely different matrices. Then the question is, I mean, how do we know the quality of the quantitative data is good? Or if you have multiple pipelines to process the data, just like the database search, right? I mean, which one should you use? I mean, I think that's still an open question. We have begun to work on some criteria. For example, you can look at the interacting partners. You would want them to be co-regulated, ideally in your data set. That may be one indicator of the data. If you compare two processed matrices, right, that may give you some idea, like all the proteins in a complex tend to be good. Then maybe that indicates this normalization or batch effect removal is actually working. But I think we should develop more benchmarking metrics for quantitative data that will not also enable us to like identify high quality data to integrate and analyze from the data repositories.
Oh, you know, I love that. Quantitative benchmarks. It's like, love it. We have two last questions. One question for Karen from actually a panelist from Nuno. Is it possible to submit reanalysis results to the Proteomics Data Commons, or is Proteomics Data Commons submissions are really only for new raw data?
I think that's a great question. I think that they could probably come in as a additional analysis of a certain study. I mean, we would need to just bring that forward, but I think that would increase the richness of the PDC, because we know that even within our own presentation, we have what we consider a common data analysis pipeline. It was a pipeline that was agreed upon by the community, but we know that when the different research groups that have slightly different expertise, for example, Bing's group or some of the other members of the consortium may run computationally different. So we actually have different sets of the data, even within the PDC presently. So I think it would be kind of exciting to have that done. We would have to just find that the best way to present it, you know, so that it would be there, but that would be really exciting. Yeah.
Yeah, I love it. And Nuno's posed a question to everyone. We have just a couple more minutes left. Kind of the thought that he's going to end with is, what is the main thing you wish repositories could do for you that can't be done today? So kind of looking forward, the future of repositories. We discussed a lot of different things, adding metadata so we can better combine different experiments, quantitative or otherwise, like spectral libraries. We talked a lot about how to reuse data that might be in these repositories, which seems like maybe one of the main things that users wish that they could do that they can't easily do right now. So we've got just a minute left. I'd like to thank all of our panelists. If you'd like to weigh in on this question, I've been kind of live tweeting as we go. I know a couple of our panelists are on Twitter. If you're not on Twitter yet, we could get you on maybe to get your thoughts from the community on this topic of data repositories. I'd like to thank again all of our panelists. Thank you so much. It was a great discussion. I'd like to thank US HUPO for organizing the discussion for everyone. And yeah, I hope to see you guys at our next virtual meeting, our next virtual panel. And if not, I'll see you on Twitter to continue the conversation. Thanks, everyone.
Thank you.